Hello, and thank you for joining us for today's webinar with Chrysalis Partners. My name is Jason Amsbaugh. I'm CEO of Samba Scientific and the host of today's talk. Today, we are joined by three speakers. Dr. Charles Chiu is a professor of laboratory medicine and infectious disease at UCSF and director of the UCSF Clinical Microbiology Laboratory. Charles is an infectious disease physician and clinical microbiologist. He also leads a translational research laboratory focusing on metagenomics-based technologies for virus detection and discovery. He has significant experience using DNA sequencing technologies for infectious disease diagnostics and discovery of emerging pathogens, including such applications such as point-of-care outbreak monitoring in remote settings and aboard the International Space Station. Dr. Brad Perkins is Chief Medical Officer at Chrysalis, a biotech company with a liquid biopsy test that uses NGS to detect over 1,000 pathogens from a single blood draw. Brad is a physician, scientist, and entrepreneur with extensive experience in microbial and human genomics research and commercialization. At the Centers for Disease Control and Prevention, he led the anthrax bioterrorism investigations following 9/11 and later served as CDC's Chief Strategy and Innovation Officer. More recently, he was Executive Vice President for Strategy and Innovation at Vanguard Health Systems, and he was also the founding Chief Medical Officer for Human Longevity Incorporated. Dr. Perkins and Dr. Chiu are joined today by our moderator, Rob Cohen. Rob is a partner and managing director of Chrysalis Biomedical Advisors, a strategic consulting group focused on high-growth companies developing innovative healthcare solutions. Rob has held various commercial leadership positions at Applied Biosystems and Thermo Fisher Scientific, as well as Illumina and Agilent. During the presentation, if you have questions, you can submit them using the Q&A button at the bottom of your screen. Please put your questions here and not in the chat window so that we can be sure to bring them up for panel discussion. Also, we are recording this webinar, so if you miss any part of it or want to rewatch it or share it with colleagues, you'll be notified in a few days by email that the video is ready to watch. Without further delay, I'll turn time over to Charles, Brad, and Rob.
Hi, everybody. Thanks so much for joining. I'll wait for Brad and Charles to come off mute. Thanks. Hi, Charles. Hi, Brad. So launching right into it, let's get an idea of how both of you got interested in microbiology, clinical or otherwise. We'll start maybe with you, Charles, and then Brad.
Sure. So my background actually is both in infectious disease and engineering, and I think my interest in microbiology began during my medical school residency, where I noted that upon seeing patients that it was very difficult to make a diagnosis. And I recall being frustrated because we have many patients who have very complicated medical histories, and they present with, in the hospital, especially at tertiary care hospitals, they present with very atypical presentations. And I think that's what drove me to develop or attempt to develop through my research to try to develop methods by which we can more accurately and diagnose infections in these patients and provide kind of more timely diagnoses and management.
Hello, everybody. Thanks for the question, Rob. Good to see you, Charles. Pleasure to have this conversation with you. I actually go way back when I was a high school student. Interestingly, my father had a good friend who is a clinical microbiologist. And one of my first jobs was to go to a slaughterhouse in St. Louis, Missouri, where they were slaughtering sheep to obtain sterile sheep blood for use in blood culture plates. And that sort of have started sort of what's become a lifelong fascination with microbiology. As an undergraduate, I did a degree in my undergraduate degree in microbiology. Thought about doing a PhD, but became convinced that going ahead and doing medical school would be a much more flexible path that would allow me to pursue both research and clinical care. And then I did my residency in internal medicine, chief medical. I was chief resident at Baylor College of Medicine, and then I was off to CDC for a couple of decades, and really off to the races with many of the many parts of the technologies we're now using at Chrysalis, and in clinical metagenomics generally.
Terrific, thanks. Well, to the topic at hand, what do both of you see, and we'll take maybe Charles, then Brad, just by order. What do you see as the limitations of current and I guess near future standard of care approaches in clinical microbiology diagnostics?
Yeah, so maybe I'll tackle this. I mean, this is a huge issue in that, traditionally what we've been taught in medical school is always to basically see the patient, and it's very important to see the patient, examine the patient, formulate, take a history and physical, formulate a differential diagnosis, and then send off testing that helps to either confirm or refute kind of your differential diagnosis, what you think are the top 5 to 10 most likely causes of the patient's acute illness or chronic illness. And so the problem with this approach, though, is that at the same time, you need to manage the patient. You have a patient who's acutely ill, who's in the hospital, who you're taking care of. And so you're really working at all times, or pretty much at all times, with incomplete information. And so I think that's the big issue, in that we really lack broad-based diagnostics. We have a very sensitive and specific diagnostics, or at least in microbiology, for specific pathogens. Or we have more broader attempts of like, so for instance, culturing or growing organisms. But they all have their limitations in that typically we're only looking for maybe either one or a handful of pathogens at any given time, whereas more than 1,000 pathogens in principle can cause a clinically indistinguishable clinical illness. And so that's why in infectious diseases or in medicine in general, we have a very serial approach towards diagnosis, where we send off test after test after test. And as a result, many of these patients undergo a diagnostic odyssey in the hospital, where we're unable to make a diagnosis in a timely fashion so that we can accurately and effectively treat these patients.
Yeah, and as I've thought about it over the years, it's many indications go through diagnostic odysseys, rare disease, et cetera. But infectious disease seems to be unique in that often cases proceed quickly and patients go downhill quickly. Is that a fair assessment?
That's a fair assessment. It depends on the disease that you're looking at, but certainly for patients who are admitted, who are acutely ill with these very severe presentations of clinical syndromes, including pneumonia and sepsis, meningitis, encephalitis, neurologic infections. I mean, many of them can get worse, clinically deteriorate very, very rapidly. And so having tests that not only are broad, but also that where the turnaround times are relative, or within an actionable time frame, become very important.
Thanks, Charles. Brad, your perspective on limitations.
Let me, Rob, let me ask if I could take a little bit different cut into the problem that we're trying to solve with our technology, Chrysalis. I'm a public health guy and sort of born and bred. So I think before we get to a potential solution, it's really essential that we define what problem we're trying to solve. And we've been very disciplined in thinking through that. And the problem that we're solving, and the place where we believe that there is a urgent, high priority need for this kind of innovation, is among immunocompromised patients. And it's really, immunocompromised states had a very important moment, unfortunately, during the COVID pandemic, but it's recognized to be a growing problem as a result of ironically, successes in a variety of medical fields, but also changing demographics in the United States. And the 2021 National Health Interview Survey, published in JAMA in 2024, indicates that about 7% of adults have an immunocompromised or immunosuppressing condition. That's over 16 million people. And that is the population that we believe is being most adversely affected by what is a very clear failure in conventional microbiology testing. And it comes in a variety of flavors. The worst aspect of that failure is diagnostic yield. And in this population, the kinds of yields that we see for these immunocompromised individuals in the United States are just desperately poor. We've done a couple of systematic literature reviews in some of these populations, those with cancer and undergoing stem cell transplantation, those with solid organ transplants. The yields from culture, which is unfortunately still the gold standard in caring for these patients, are down in, over the last decade, the high quality studies that we looked at, the yields for culture are down in the 20 to mid 20% ranges. And that generates a huge problem with clinical care for these patients. It's not only the yields, it's also the lack of ability to detect uncommon and fastidious pathogens, the fact they're on empiric therapy for long periods of times that drive resistance and have adverse events, the fact they're getting invasive procedures when plasma-based microbial cell-free DNA testing could avoid many of those. And then, as Charles stated, the state of the art is to use these small panels of PCR testing or single test in successive waves of testing that really never arrive, very rarely arrive at a clear diagnosis. So many of these immunocompromised patients never get a diagnosis. And those that get a diagnosis usually get that at a time that's so late that they've had, tremendous healthcare complications. I think of this, in my public health perspective as an incredible failure of our current microbiology testing practices. And this is what I think about and what our entire team at Carius thinks about when they get up in the morning. And so that for me is the problem we're trying to solve right now, at least at Carius, with this technology.
Thanks, Brad. With that, Brad, why don't you tell us a little bit about the approaches that you're using at Carius and developing, and then I'll ask Charles the same kind of these are the metagenomic type approaches, or at least multi-pathogen approaches.
Yeah, so thanks for the question. Carius, we think of as a global pioneer in a particular flavor of clinical metagenomics. And we talk about it at a high level as a liquid biopsy for infectious diseases. We were founded in 2014, out of Steve Quake's laboratory at Stanford University. Importantly, we were founded out of the tradition of other cell-free DNA applications in clinical medicine. So non-invasive pregnancy testing, uses in cancer, uses in organ health. In the context of infectious disease, what we do is actually a little different than some of those other areas. Is that we leverage a single analyte, which is microbial cell-free DNA that is found in plasma for both the diagnosis and management of infections anywhere in the body. And it's it's like one of the greatest tricks, in science and technology right now, that you can use this analyte that's harvested from an infection anywhere in the body to detect that infection. And very importantly, we do not focus on or need or even want intact microbial cells or intact viral particles in our assay. And this this is a particular flavor of clinical metagenomics, but it's one that we've spent a decade at Carius working through the challenges in the technology, which are numerous. The two major challenges, and what makes this sound easy but is actually quite hard, the two major challenges are enhancing the signal of microbial cell-free DNA against a very prominent background of human cell-free DNA. And then the other major challenge, can sound trivial, it's not, I assure you, is handling the ubiquitous environmental contamination of everything with microbial DNA. And that includes all the reagents, and all the handling of those specimens, and we have repeatedly innovated in both of those domains over the last decade.
Fascinating, thanks. Charles, some of the approaches that you've been using over the years, I know that they are many and varied.
Yeah, so that is true. They're quite diverse, but I think in general, we also adopt essentially what we call a metagenomics approach. So the whole concept behind metagenomics is that in principle, nearly any type of infection, you're probably only missing prions, which are protein-based, but nearly any other type of infection, whether it's bacterial, fungal, viral, or parasitic, could be identifying by identifying DNA or RNA. So and for RNA would be needed for RNA viruses. But the general idea here is that we want to be able to implement what we call an agnostic test, meaning that it doesn't target any one pathogen or even a group of pathogens. It simply sequences, it does shotgun sequencing of DNA or RNA from clinical samples. And it's a very attractive approach, as Brad alluded to, simply because it's completely hypothesis free, in that you can see simply running the clinical samples, shotgun sequencing of the nucleic acids, and then trying to figure out, can you identify non-human reads that may correspond to pathogens, whether bacteria, viruses, fungi, or parasites. We tend to we also do cell-free DNA and RNA approaches, but we're also interested in cell-associated DNA and RNA because the other advantage of doing sequencing is you actually generate a huge amount of information that you potentially could leverage even beyond looking for a pathogen. To give you an example, we're currently leveraging RNA to be able to identify gene signatures, because we're also sequencing the human RNA, the human host RNA that's made by the patient in response to the infection. So presumably, all of this information is complementary. And so the idea is, and as I believe that Carius has also been interested, for instance, in further mining, for instance, the bacterial sequences to look for like antimicrobial resistance, for instance, and so this type of information can kind of go beyond simply identifying the presence or absence of a given organism. The other thing also that makes this approach very attractive is that once you develop a quote metagenomic protocol, in principle, because you're not using specialized or specific primers or probes, you're not targeting any one or group of organisms. Because of that, you can essentially apply your protocol across not only a single body fluid or type, but you could also extend it across potentially many different body fluids or even tissues. And so we've started primarily with spinal fluid. I know that Carius has started with blood or plasma, but in principle, you can extend this towards other body fluids or towards other tissues. And that becomes very, very important because some infections can be localized. Some infections might be, for certain types of infections, there may be, a preferred sample type, because usually the infection may be concentrated, for instance, in a given organ, or may be sequestered or compartmentalized. And so that's why I feel that the flexibility of this approach and the fact that it is hypothesis-free makes it potentially a very powerful tool that we could use in the clinical setting.
Charles, talk a little bit about a patient maybe that's a success story that benefited from an approach like the one you've talked about.
Sure, sure. I can talk about. We have several cases. Maybe I'll talk about. We had a 10-year-old girl who was from Mexico, who presented with essentially headaches, and then developed a stiff neck, and then had some was a little altered, was confused, and was admitted to the hospital. And essentially, she was admitted to the hospital for several weeks. And this is in terms of getting a workup. You know, all of the diagnostic testing was negative, and it was at that time that we actually, this was several years ago, even before we had any clinical metagenomic testing. And so she was eventually diagnosed with likely TB, tuberculosis. And one aspect about tuberculosis is that it's incredibly difficult to make the diagnosis of tuberculosis because typically, typically when you the bacteria is present in very low titers, so it's very difficult to culture. It can be fastidious, so difficult to culture. It may be difficult to detect by polymerase chain reaction, PCR-based methods, and often in cases where we suspect clinically that a patient may have tuberculosis, we may treat empirically, meaning that we would treat without having an actual diagnosis. So she was thought to have likely tuberculosis and was then treated with 4 drugs, and did get initially better after several weeks of treatment, but then came back to the hospital because she continued to have symptoms. She was continued to be intermittently confused with headaches, and was developed, essentially had nausea and vomiting and weight loss. And so came in with persistent symptoms. And it was at that time that we then enrolled her in a research study that would allow me to run a clinical metagenomic test. And we actually found out that she indeed did not have tuberculosis. She actually had a different disease called brucellosis, which is a different bacterium. And it seems like she probably got exposed to this disease because she was, her family had been eating raw cheeses. And so one of the transmission of this particular disease is through raw or contaminated food. And so she was diagnosed with brucellosis and was then put on appropriate treatment. And it had turned out that she had initially gotten better simply because some of the drugs that are used to treat tuberculosis overlap with those used to treat brucellosis. So she was effectively being partially treated, but not fully treated. Yeah. And I think it really highlights, and she did get completely better with the appropriate treatment. Right. But I think this really highlights the importance of making an accurate diagnosis. But it also highlights kind of the difficulty of making a diagnosis in these patients. She was admitted to the hospital and was being worked up for about 2 to 3 weeks. And yet, even after 2 to 3 weeks of extensive testing, they were still unable to, clinicians were still unable to diagnose her disease. Yeah.
So is it fair to say in the absence of a metagenomic test, they would never have found, or it would have been difficult at least to find?
Yes, I think it's unlikely that they would have found it because they did not entertain the diagnosis. And I think that's an important, I think a very important point to make, in that it's not that we don't have tests for these diseases in several cases. We probably do have tests for these diseases, but it happens to be one of perhaps hundreds, in cases thousands, of potential reference tests that you could send out. And you never have enough clinical sample, and you never have enough, you don't have the ability to test for all of these pathogens at once. And so that's why I think that, you know, having the ability to test simultaneously for thousands of pathogens becomes very attractive. It's also a very economical use of kind of limited resources that you may have to work up and diagnose these uncommon infections in patients.
So, Brad, where are we on the adoption curve of these kind of cell-free approaches that you talked about? You can take, NIPT, cancer, organ health, and maybe infectious disease, and generally kind of paint a picture of where we are.
I think we're if you look at the adoption of other cell-free DNA based tests, I mean other clinical indications, I think we're on track. You know, the earliest, I pulled some data looking at, for each of the major cell-free DNA clinical domains, when was the first research published that suggested a potential clinical application? And then when was the first inclusion in the first US professional guidelines? For cell-free DNA, It was sort of out of the box early 1994. There was a paper published, and then in 2018, there was a U.S. professional guideline to identify, EGFR mutations for treatment in adenocarcinoma of the lung. That adoption cycle took 24 years. Okay. NIPT was a little faster. It got into guidelines, U.S. guidelines for high-risk pregnancies in 15 years, and then it was recommended by U.S. professional societies for all pregnancies in 23 years.
Yeah.
Oregon Health application is the clear winner. First research was 2011. And the first guideline, it was a limited guideline, but it was a guideline for use in kidney transplants in 2017. It took 6 years. So if I do that same, if I mark those same milestones for microbial cell-free DNA, Steve Quake published the first research paper in 2015 suggesting the application we're now using at Chrysalis. We are not yet in US guidelines. We do have one sort of international guideline in endocarditis for the use of our test. But we're still we're looking for our first US professional guideline. I believe, as objectively as I can, that guideline should be coming very shortly in the use of our technology for diagnosis of invasive fungal diseases. But so if you say that we'll have a U.S. professional guideline in the next year, that would put us at 11 years. So my diagnosis, which I think is different than Charles, is that we are on track. We are seeing strong adoption with the use of our technology among immunocompromised patients.
Yeah.
And I think that's really important because it's about what problem you're trying to solve.
Yeah.
That's the problem that we're committed to solving. Our stewardship is all about that. And then there's a practical issue around evidence generation. Okay, all this, professional guidelines and physician adoption is driven by evidence.
Yeah.
You can't boil the ocean. I think this technology is incredibly powerful for many patients, but you've got to be focused in your development of evidence, and we are very focused on serving what we think is the most urgent need for this technology in immunocompromised patients.
Yeah, I like that idea of focus. Charles, your perspective contrasting on kind of where we are in the adoption curve, and then maybe kind of thinking about other types of infections that can benefit, or other patient populations that can really benefit from this beyond immunocompromised.
Yeah, so I don't think I'm that far off from Brad's assessment. I believe that we're sort of like in the middle of this, in the sense that we're clearly not there yet, and that I mean there isn't yet a kind of a well-defined indication for any of these tests. And yet, I think that there is a roadmap, in the sense that we now have enough experience overall with the whole field of clinical metagenomics that we know where the likely indications are to be. And so one of them is indeed immunocompromised patients, so CASHA recipients. Not only bloodstream infections, but potentially as Brad said, you can survey the blood as a way to look for infection elsewhere, but also immunocompromised infections in other settings. So, for instance, in respiratory disease. So I think some areas of where of clinical unmet need include, for instance, more accurate diagnoses of pneumonia, especially atypical pneumonia from various bacteria, fungi, and parasites. Diagnosis of central nervous system infections is meningitis and encephalitis is a huge problem. Even today, roughly about 50% of these cases of meningitis and encephalitis do not get a diagnosis or do not get a definitive confirmed diagnosis for hospitalized patients. The other thing that makes, I think, neurologic infections quite interesting or quite distinct is that the differential diagnosis is very broad. And in fact, it may be more non-infectious etiologies of meningitis and encephalitis actually may be more common than infectious etiologies. So it's not only microbiologic testing that you in principle need to do. You would need to test for autoimmune disease, for vasculitis, for neurodegenerative diseases, for sarcoidosis, amyloidosis, unusual autoimmune syndromes. I mean, these are all non-infectious or presumably non-infectious conditions that can mimic an infection. And so it poses a huge issue. And so having again, a metagenomic test where you can deal with the infectious causes, but importantly, we're actually trying to investigate the potential of a metagenomic test for ruling out infection. And that becomes important because for neurologists, for instance, or infectious disease physicians taking care of these patients, very often the decision is, does the patient have an infection where you would want to treat with a targeted, antibiotic or antiviral versus does the patient not have an infection, perhaps with an autoimmune disease that is mimicking infection, where the treatment is actually the other way around. You're actually treating with immunosuppressive agents, such as steroids or immunomodulatory agents, which you probably would not do if the patient indeed had an infection. And so being able to kind of use a test almost as a soft rule out test as a way to potentially rule out infection becomes very intriguing, and it might be a specific application. I think another area is in and we've sort of only touched on this, but is in the area of emerging infectious diseases and in the setting of surveillance, for instance. And so this is where diagnostic medicine crosses with public health surveillance. But it is true that we have continued to encounter essentially novel viral infections, emerging viruses, emerging pathogens. Several instances include that this month, for instance, we're publishing a manuscript or a paper in Emerging Infectious Diseases that describes two novel Bunya viruses that were described as causing fulminant encephalitis in immunocompromised patients. And what's interesting about these two Bunya viruses is that these two Bunya viruses had never before been detected in human samples. They were actually detected by mosquito and tick-borne surveillance. So they're presumably mosquito viruses, or they harbor mosquitoes and ticks. And there was a zoonotic transmission that caused an infection in these two individuals. So the use of, again, a very broad technique to aid in potentially emerging infectious diseases in the future is also another potential application.
That's great. Thanks. Brad, you touched on evidence generation, and it's a complex topic, and obviously we have a lot of complex topics and don't have the time to address them all. But just in general, how should we think about where we are on the curve of generating evidence for, let's say, payers? Obviously, you can split payers into public and private, but kind of where are we and what needs to happen?
Yeah, I believe that we're at a very exciting inflection point, and I would differ a little bit from Charles, who said that the applications aren't clear. I think actually we have remarkable evidence for a number of really important clinical applications for our technology. Those include endocarditis, pneumonia, invasive fungal infections, which is sort of a subset of the pneumonia, febrile neutropenia, and fever of unknown origin. Carius has now, we just crossed over 200 peer-reviewed publications, and many of those are reasonably high quality. We have 23 prospective studies, 46 retrospective studies, 15 case series, 106 case reports, 2 budget impacts and cost effectiveness analysis. We feel, in interacting with a number of influential payers and others, that the evidence now is starting to become overwhelming for the clinical use of this technology. I would cite two areas in particular, endocarditis, where we already in 2023, we were included in the Duke criteria and the International Cardiovascular Infection Society disease recommendations. Primarily, and this is to Charles' point, which I'm very attracted to, but again, we're trying to solve a public health problem here. You know, we're included in the endocarditis recommendations to really, because of our ability to detect three particularly fastidious but relatively common pathogens in endocarditis. Bartonella henselae, Coxiella burnetii, and Trophirma whippleii. There's no commercial test for any of those that are really useful in endocarditis. We detect all three reasonably well. It's fun for me, particularly if I'll digress a little bit, is I led the team at CDC that identified the cause of cat scratch disease and published it in 1992 using 16S PCR primers. And Bartonella henselae is the cause of cat scratch disease, and it's a very common previously unappreciated infection in many immunocompromised hosts. So it's gratifying to see us identify a case with that infection almost every day. So endocarditis, I mean, it's sort of a slam dunk. I mean, physicians that are not using this technology for endocarditis, my heart is breaking for those patients and families, because this is an extraordinarily serious disease among solid organ transplant patients in particular. The rate of endocarditis is 170-fold greater than it is in the general population. So endocarditis is the evidence is really superb. The other thing I would say basically in pneumonia, and particularly in invasive fungal infection, if you are immunocompromised and you have a lower respiratory infection that requires hospitalization, I mean, I really urge any physicians or people at health systems to take a look at the data that we've published, because it's quite extraordinary for pneumonia. And it's really important because, again, public health, my old colleague from CDC is at the Institute of Global Health and Health Metrics in Seattle. They publish all the definitive burden of disease data. In 2018, they published the most recent assessment of burden of disease in the United States. The only infectious disease on their list of top 25 years of life lost is respiratory infection, primarily pneumonia. So this is a huge problem in the general population, like Charles said, but it's particularly desperate in the United States. One of our largest studies, the Pickup study, it was an observational study across 10 of the largest cancer centers in the United States, but in hematologic malignancies and stem cell transplant. The mortality rate was 25%. And the number of those folks that were diagnosed by standard of care, including bronchoscopy and BAL, was just miserable. The Carius test increased that rate of diagnosis by 40% in this study. So my hope is that people will become more aware, and more lives will be saved, and more families will have less suffering as a result of the use of our test with pneumonia, particularly in fungal disease. And I think this will resonate with Charles too, but that if you just take a look at it, because it's not only yield that we provide to clinicians with invasive fungal disease. We provide physicians with a quantitative assessment of microbial burden across all of our detections, but this is particularly important in fungal diseases because it allows physicians to monitor the use of antifungal therapy.
Yeah.
We've demonstrated, because of our increased sensitivity and improved yield, we've demonstrated, I think, quite convincingly that we can, in high-risk patients, we can identify invasive fungal infections before they become clinically apparent, which is sort of mind-blowing. We have not had technology to be able to do anything like that. We can speciate, and Charles knows this very well, the conventional microbiology testing does a very poor job in speciating invasive fungal infections, and much of the antifungal therapies can be better selected and targeted when that species is available, particularly for Aspergillus. And then we know from a couple of studies that the quantitative fungal loads in patients can be used to predict mortality and/or extrapulmonary spread, and this turns out to be much more common. It's completely impossible to do this with conventional microbiology testing, but we can identify multiple invasive fungal infections caused by different taxa simultaneously with our technology. That's virtually impossible in any clinical setting without this. Although all those things seem very basic in their clinical importance. So back to your evidence, we're not as far as we'd like to be, but we have the momentum is building for national guidelines and for payer inclusion.
Yeah, I'm thinking that's a great overview. Thanks, Brad. I'm thinking about where we are on the HEOR realm. So in the health economics and outcomes research realm, and the things that payers, of course, at the end of the day, because it's a business, eventually look at. Where are we on that? And Charles, do you have a perspective on that?
Yeah, it's actually a really interesting question. And I think that in some ways, it's almost as if in some ways, we're sort of getting ahead of ourselves, is my feelings. Because I want to push back a little bit on this in terms of like exploring the question of, it's great that, for instance, that, like the Curious test is now on Duke's criteria, arguably it should be used. And I actually feel that, we should do this type of testing for all cases of culture negative endocarditis, for instance. But the problem is with that, or one potential issue with that, is that because it's already being used as part of clinical practice in many different settings, it becomes very difficult, for instance, to then develop essentially a randomized controlled clinical trial. I mean, usually we consider randomized controlled clinical trials as kind of the gold standard, where you have the intervention, where you do the test, or where you do not do the test. And arguably, for certain indications now, it's almost unethical not to do the tests or to withhold, say, the testing on a certain group of patients. And so my feeling is that moving forward, I think that it may be important for us to think in advance in terms of exactly what kinds of prospective clinical studies. What would the design of a prospective clinical trial be in such a way that would convince, say, payers, and also would produce results that would be robust, that would really enable us to identify potentially new indications. And I think there is still, some for several indications. For instance, we don't have, it's not conclusive yet. We don't know for sure, whether or not for this particular indication that testing is going to be indicated. And in that case, then you can envision developing essentially and going through with a prospective randomized controlled clinical trial. And so that's an interesting point that I feel that's been sort of largely ignored, that essentially we're just going into this in many ways, much of the evidence that we collect, it ends up being, anecdotal because, the data is, you can do a prospective study, but it's not doing a randomized controlled clinical trial for many of these indications becomes quite challenging. And so, and the question may also be, is that the sort of evidence that, would be needed? Are those the sort of health economic outcomes measures or metrics that would be needed to convince payers to actually pay for a test like that? I'd be interested in hearing from Brad about his viewpoints in terms of like the necessity for these types of trials, which can be quite expensive, as Brad is certainly aware of.
Yeah, just before you answer, Brad, and certainly we'll give you airtime, we've got a lot of great questions from people, so I just want to leave a little bit of time to at least start to hack at a few of them. Go ahead, Brad.
Just very quickly, we have done a health technology assessment of our test with pneumonia among immunocompromised hosts, and it looks very favorable in terms of typical thresholds for disability adjusted life years. And I would love to continue the conversation about evidence, Charles. I share your ambivalence. I do think that just the structure of diagnostic, new diagnostic economics in the United States, it's very hard to afford those kinds of studies early in the development of these technologies.
Yeah.
And you may be right. We may be at this point increasingly reliant on sort of large real world data sets to get the data we want. I do think for payers, we are going to be able to get some variables around cost savings as a result of admission when we get to outpatient use of these technologies. Yeah. We will get to cost savings from procedures avoided, from antimicrobials avoided, from other diagnostic tests avoided. I think there's going to be a package of things we can put together that will convince that, particularly in these immunocompromised patients, that this is a good thing. This is a win for them. It's a win for the patients and families, and it's a win for health systems.
Is that some sort of kind of larger public-private effort? And boy, how does that get funded? There's all sorts of questions I have.
Charles can answer to that. I'm not aware of any sort of
not that one exists, but is the solution to it that? Yeah. Sorry.
Yeah, we don't have a solution to that, certainly, at this point, because I'm unaware at least of a kind of a nationwide concerted effort to do this, or even within, it has been something that has been discussed. We discuss this all of the time in terms of, but largely, unfortunately, this field has not coalesced to the point that working together towards that. So it's rather been that you have individual groups or individual companies or individual laboratories are essentially, developing, validating, and trying to look for. So my feeling is that this would definitely benefit with some sort of synergy or collaboration, basically. But it's something which is still to be explored. But it's going to be an issue that's going to be facing all of us, I feel, very shortly in the next few years.
Yeah, fascinating. Okay, in the interest of time, I want to cut over to a few of the questions. So the first is on, and it's a set of questions, but it's on targeted versus hypothesis-free approaches. So while targeted methodologies have been predominant, could there be merit in grouping pathogens by their biological characteristics, such as categorizing RNA respiratory viruses together? Charles.
So I'm unclear about that question. What does that mean? You mean targeting certain groups of pathogens, or so more targeted methods to look for those groups?
It's about batching together approaches. So I'll read it again. While targeted methodologies have been predominant, could there be merit in grouping pathogens by their biological characteristics, such as characterizing RNA respiratory viruses together? So I think the question is creating a semi-hypothesis-free test for all RNA respiratory viruses rather than being And I suspect that's being done.
That's a great question. One thing I should mention is that we actually, to some extent, are already doing that. So, for instance, we're already separating, for instance, our libraries into DNA and RNA, where we're using the RNA primarily for RNA viruses. I mean, we do use the RNA, for instance, for in some ways to support the DNA results, such as, for instance, by looking at RNA from, say, a bacterium, you can see evidence that there's active, that perhaps that it might be live bacteria, it might be active, basically an actively replicating organism.
Yep.
Whereas for RNA viruses, we really need to look at RNA libraries. So there would be some utility potentially for just doing RNA sequencing as a way to look for RNA viruses. The other benefit of this more targeted approach is that it becomes perhaps more straightforward to submit regulatory application because one of the big issues involving for instance, regulatory approval from agencies such as the FDA is that traditionally they're more used to panels, whereas you have a discrete list of targeted organisms. They're well-defined criteria. You need 20 positives of this organism, 20 negatives, et cetera. The same criteria do not apply when you're talking about 1,000 different pathogens. And in fact, not only that, but 1,000 different types of pathogens. I mean, viruses, bacteria, fungi, and parasites are quite different in terms of genome size, morphology, characteristics, et cetera. And so there might be some argument to a more narrowly focused or slightly more narrowly focused targeted approach such as just, say, RNA viruses, for instance, or in particular respiratory RNA viruses. That might be more straightforward to undergo regulatory scrutiny.
Yeah, makes sense. Next question. One of the challenges in metagenomics as a diagnostic tool is the low pathogen burden in these samples. And I think one or both of you certainly touched on this. Linking the etiological agents to the disease is highly dependent on the expertise of the interpreter of sequencing data. What are your thoughts on how to standardize the process? And I think this is obviously you running your own lab and you as directing effectively, I think, a service organization probably have different perspectives on that. Brad.
It's a great question. I'm really excited to continue to bring clarity to more clarity to the interpretation of our results. I will say that we take this issue quite seriously and are looking to bring more science to it. Just as a general overview, we do categorize our pathogens on our report into obligate and opportunistic. And then commensal pathogens and reactivating sort of DNA viruses. And that helps people, I think, interpret and use the information. We provide some context for commensals, which may be causing an infection or may not be causing an infection. We provide sort of a look at the quantification of their specimen in comparison to our historical experience with concentrations of that organism. I think that's really good. Talking to regulators about all this is going to be important. I would give as a context, and I'm looking, for input, is we think about our report as having, we have clinical infections on our report. We have that need to be treated. We have subclinical infections that may not need to be treated. And then we probably have a set of mechanical translocations of organisms or cell-free DNA alone that is picked up by our new microscope, if you will. And so this is a new view of the world, and we need to collect science and support our physicians in interpreting it in the best interest of the patients they're caring for.
Charles, your perspective on this, and then I apologize, we're getting toward the top of the hour, and thank you very much to everybody for all of the thoughtful questions. It may necessitate a follow-up. Anyway, Charles, your thoughts on that?
One thing that I've been a very strong advocate of is this concept of a clinical microbial sequencing board or a clinical consultation service. And I know that Carius has one as well. And so the idea is that if there are unusual findings, or even if there are not common findings, the idea I feel that the information is far more valuable if it's put into clinical context. And so to give you a concrete example, Staphylococcus epidermidis, for instance, a Coagulus negative staphylococci, in the context of otherwise healthy patient, it might very easily be a blood culture contaminant or blood contaminant. But in a patient who has, say, a prosthetic joint, it could be the cause of a prosthetic joint infection. So the same organ can be an infection, the same organism can be an infection in one patient, but rather be a contaminant or a colonizer in another patient. And so that's why we support all of our clinical testing. We support, we support the physicians with this clinical microbial sequencing board called the CMSB, or we call it clinical consultation service, where physicians can sort of discuss their patients and we can discuss the results and put them in clinical context. And, that might be something that might be also, part of, in terms of a regulatory submission, it might be part of, inclusion of that might actually be quite important with regards to, ensuring that these tests are, not only accurate, but also safe and effective.
Thanks. I think we have time for one more question. And we talked about, certainly both of you have touched on endocarditis. Endocarditis is an exciting high-value application. In the future, what do you think the highest prevalence indication will be that can be addressed using hypothesis-free approaches? We've kind of talked around a number of indications. So whoever wants to take that first.
Pneumonia.
I would say respiratory disease or pneumonia if it's the highest prevalence. In terms of which would have the most impact, that might be different. I would say probably CNS infections, culture negative endocarditis, infections in transplant recipients or immunocompromised patients.
Okay. Well, that brings us to the top of the hour. Brad, Charles, thank you so much for all of your time and your thoughtful answers. And thank you to those of you in the audience who stuck around, and to those who formulated all of the good questions that unfortunately, due to time, we couldn't answer. So thanks again. We love talking about these kinds of subjects at Chrysalis, and spend a lot of time thinking about these and how indications and technologies intersect, and all of the challenges and opportunities that they present. So thanks again. Hope you enjoyed it.
Thanks, Charles. Thanks, Rob.
Thanks, everyone.